Hey everyone, Connor Wander here with the Straight from a Scientist podcast. Super excited to bring you this episode because last but not least, we have Dr. Harry Steinbusch from Maastricht University in the Netherlands. He was actually the founder of the AD Fast Track Conference, which most of these podcast episodes have been from. And he's really the one who put it all together. He made it happen a long time ago, and he's still making it happen today. And he's been the driving force in getting students, postdocs, young faculty together, everyone who's researching Alzheimer's disease, and also provoking people to think differently about the disease. Now, Dr. Steinbusch's research areas vary widely, and he's extremely creative, and he's always thinking of new research topics. But what he mainly hit on during the conference and what we cover in this podcast is experience and how that changes the brain, and how that can change the brain in Alzheimer's disease. So you may be familiar with the term of epigenetics. Our previous podcast with Dr. Tunbridge discussed not epigenetics rather, but genetics and the base pair. So what is the base structure? What are the building blocks of DNA? However, Dr. Steinbusch's research studies epigenetics, kind of like what are the little modifications to the building blocks of DNA. So what are the colors of paint on the building blocks of DNA that allow it to be read or not to be read properly by the cell? And we think that all sorts of dysregulation happens. It's all mucked up in Alzheimer's disease. But the good news is that some of the things that people do in everyday life can change the way epigenetics modify DNA in the brain and can change the birth rate of adult neural stem cells, which are very important for regenerative purposes, but also just learning and memory, which of course are impaired in Alzheimer's disease. So I think it's a great recap for all of these episodes as a whole. We've talked about things like sex differences in Alzheimer's disease. We've talked about neuroinflammation in the disease. We've talked about disease progression, and I highly recommend you check out all of these episodes if you missed any and you're curious. You can find that actually at the top bar of our website. It's the last link in the menus, the focus on Alzheimer's disease, since we've just had so much great discussion by all these great experts on what the disease means to them and how they think we're going to cure it and what they're working on, how they're driving us forward to that. But anyways, I'll let Dr. Steinbusch get to it. It was an amazing episode, and I really have to thank him for being so patient with me and getting this all going, because we had a lot of hit and misses in making it work overseas and whatnot. I hope you all enjoy, and while this won't be the last topic on Alzheimer's disease that we do, it does wrap up our series. If you have any feedback on this long series and any further questions about the disease, we'd be happy to answer them. I think we're definitely due for a roundtable on the disease as a whole, and that'll kind of tie everything together really finally. I really want to thank Rachel and Martha from the Bright Focus Foundation. I couldn't have done it without them, and they helped me organize the show notes as well. So huge shout out to them, and of course the Bright Focus Foundation, everyone involved in putting on that conference. I want to make one more quick announcement. We have officially launched the Straight from a Scientist network, the SFS network. You can find that at straightfromascientist.com/SFS network. This is basically just a grouping of scientists, blogs, other podcasts, and just general tools and resources for people who are scientists or science interested, wanting to learn more about the field. Maybe you're a student trying to find that perfect fit for a research interest. You don't even know where to start. So head on over to the SFS network. We have a great grouping of scientists who we've worked with or plan to work with in the future. And if you're interested in joining, please email us at straightfromascientist@gmail.com with a bio picture, as you see in the thumbnails, something like that, and a little preview for whatever you're working on. And of course, any social media links or places you want to forward people. And with that, give it over to the episode. Enjoy, everyone. Welcome everyone, Connor Wander here. online interview with Dr. Harry Steinbusch. Unfortunately, both of us were so busy during the conference that we didn't get a chance to talk to each other and sit down at the actual Fast Track conference. So this is going to be an add-on we're doing over the internet. Dr. Steinbusch has also done a ton of traveling, so maybe we'll talk about that later in the episode. First off, Dr. Steinbusch, thank you so much for coming on the podcast.
You're welcome.
So you're a professor in cellular neurology at the Department of Psychiatry and Neuropsychology at Maastricht University in the Netherlands, right?
Exactly. I'm chairman of the department.
Now, you do also wear a lot of other hats. Some of the things I saw from your bio online, you're president-elect of the Neurotoxicity Society, adjunctive professor at Kyoto Prefectural University, which is in Japan. You have positions in Sri Lanka, and you told me you were going to Malta, South Africa, and of course in San Diego, where we met. You were traveling there as well. How do you manage having a lab with all of this stuff going on?
Having a lab means that you have good associates who are working with you, who likes to work with you, who are loyal to you, and who really like and are enthusiastic about the fact to work together. So I do have some good people working with me at Maastricht University. They handle our PhD students, they handle our postdocs, and that's the way we're going. But basically, I'm in touch with my lab all over the world every day. So I always take about half an hour or an hour to talk with my people in the lab and say what's happening, what's up, and I got the latest information. So that's basic work, and I also encourage my own people also to travel. Travel is not just travel; travel is networking. So I'm a real networker, I could say that.
And that definitely showed at the conference. I saw one of the reasons that you were so busy was you made a very personal attempt to interface with, it looked like every single person who attended the conference, and I really appreciate that. And I think it's been seen by all the people who I've talked to. Oh, sorry.
Well, the idea was basically that I really would like to have a personal contact with each of you. You made your elevator pitches, so that basically shows who is doing what. And basically afterwards, I thought the important thing of this meeting is also that you learn to network, that you make that it must be as easy as possible to contact me, and that's why I decided to contact each of you and to have a very small personal note with everybody of you, all the students.
And when we talked, you asked me a bit about my side interests, which were the connection between mood and activity, lifestyle, and neurodegenerative disorders, which of course were the subject of the conference. So in the context of Alzheimer's disease, I'm very interested in the comorbidities with depression. So a lot of people think depression opens you up to neurodegenerative diseases like Alzheimer's. How do you think that treatments for depression might have some sort of insight into Alzheimer's disease?
Well, that's a very nice question to start with. And basically, it's one of the emphasis I put at this meeting and which we're also going to put at the next meeting in Chicago. So we like to talk about comorbidity. And a lot of people is if they talk about Alzheimer's disease, they think always about Alzheimer that has something to do with memory, that has something to do with hippocampus, that has something to do with amyloid and tau pathology. But basically, they forget that it's a multi-system syndrome. And I like to look not so much to animal models. I prefer to look to a patient. And that's why I always ask the students, who is familiar with an Alzheimer patient, who has a grandfather or grandmother who is Alzheimer, and have a look to them very careful and personal way. And I will see in our hospitals Alzheimer patients, and we know that about 80% of the Alzheimer patients also suffer from depression. And basically in the past, that depression was never treated because everybody tells you, well, if you have Alzheimer, you should be depressed because that's a major event that you have. Nowadays, we know that if we have early diagnosis and we can treat depression at a very early age of Alzheimer's, so basically stage 1 or stage 2, or Braak stage 1, Braak stage 2, then the progression of the disease is sharply reduced. So treating the comorbidity, treating depression, has a profound positive effect on the decline of Alzheimer itself.
Wow.
And that has also been tested in animal models. And of course, there the people look, if you treat an animal with an SSRI, will that result in a less rapid buildup of amyloid and tau pathology? And it does so. We know, of course, also that this pathway is going through BDNF, the brain-derived nerve factor. So basically, antidepressants have an effect on an upregulation of trophic factors. These upregulation of trophic factors has then an effect on the production of progenitor cells in the hippocampus, and by that, diminish the decline in Alzheimer's disease. So that's a major thing.
Yeah, it seems to be a really strong connection, and it makes me worry about, at least in the United States, where elderly people, especially those with dementia and Alzheimer's disease, are put into care homes because, of course, it is extremely time-consuming and difficult to take care of some of these people. But those places always seem to be devoid of much stimulation. And I know BDNF, neurogenesis are very closely tied to environmental enrichment. It's a common experimental paradigm that people use with animals. And I just wonder how, or if I can get your take on environmental enrichment in the real world. Now, I don't know exactly what that might be, but I would think an old folks' home is kind of the opposite of an enriched environment.
Well, you're making now some assumptions, and I really would like to correct them.
Sure.
The first thing is, and a lot of studies has been done, and I really would like to focus on the study from Henriette van Praag. It's a Dutch name, but she is an American, and she works nowadays in NIDAR, in the National Institute of Drug and Addiction in Baltimore. She looks, together with Carl Gottman from the University of Irvine, she looks to what you called, and what I would call also environmental enrichment. And she really looked carefully in her animal studies and mentioned what is this really environmental enrichment. There are two factors. So you have these animals, or your rats. You can bring them together. You can say you have 8 rats together, 4 males and 4 females. And first of all, in the environmental enrichment, it means that the animals by itself will start to have a social hierarchy. But the other important thing is, if you look in these environmental enriched cages, the essential factor what's happening is in fact the treadmill. It's really the exercise. It's really the rats who jump on the treadmill and walk for 4, 5, 6 hours on the treadmill to have the activity. So the environmental enrichment can be brought to physical activity. Now, there are two interesting things to mention. So we know now that if you do physical activity, your BDNF levels are going up. And then the next question was, which was done by a big study in Sun City and near Phoenix, Arizona, but also in Germany recently, is that they said, if you look now to elderly people, and we put together groups of 20 elderly people with mild cognitive impairment, and we said these people, they do physical activity, or something like we can say they used in the different apparatuses like cycling and the whole thing, or they do a grouping together. And in Europe we call it Pilates, and I think you do the same thing in the United States as well. It's in the gym.
Yeah, I've seen that.
Basically, it was shown that the people who do Pilates, so the group activity, was much more effective, twice even effective, than people who do physical activity without any social interaction. So just doing the 10 machines the 10 machines 10 times, that is much less effective. So if you would say what is effective, physical therapy in a group combination is the most effective therapy. And if you then look to elderly houses, and yes, we also have these elderly houses in Europe and also in the Netherlands, the same like in the US. And they can be sometimes a little bit, I would say, depressive and not really rewarding. But at the moment, we really stimulate elderly people to do activities. We call it biodanza here in the Netherlands. And I think you will have the same thing. I know that from my colleagues in California. I'm not familiar with North Carolina, but I think this is coming up more and more, that you really say physical activity in a group is a rewarding therapy to prevent the further breakdown of your brain in Alzheimer's disease.
Yeah, so it seems like there are two components, or, well, for sure more than two components, but it seems like there are multiple components in this environmental enrichment paradigm, which some people call like a Disneyland for rats, is another way to explain it. Thank you for your insight on that. I really appreciate it. And maybe we can throw some of the citations in the show notes afterwards so people can reference them. Would really like that. So it does seem like how you live your life and your risk for Alzheimer's disease are tightly linked. Are there any things that you do in your life that you think research has taught you are beneficial?
Well, that's a little bit more different question, a little bit
right.
Question. So, can you phrase it in a different way?
One of the questions I always like to ask is, what are some of the things you've learned from research, and how have you applied that to your life? So, one thing that is actually relevant to, I think, your recent interests in the locus coeruleus, as I read on your ResearchGate profile, is that the noradrenaline release seems to be very important. Now, I don't know that this is necessarily the right way to go about it medically, so I'm not advocating that anyone do this, but I have heard that cold showers induce norepinephrine release from the locus coeruleus, and I've taken quite a few cold showers. I also find it exhilarating in a sense. So I'm wondering if you've made any adaptations in your life based off of research results that you've seen, perhaps with exercise, switching to group exercise maybe.
Well, basically, I do not take cold showers because I hate cold showers. It's the first thing.
I don't blame you.
But yes, I do. I take something which I learned from my studies, which I learned from my colleagues. I like to take in practice. And the first thing, very practically, is I decided a couple of years ago to stop driving to my work, and now I cycle to my work to have my direct activity, my daily activity, is to really focus to say I'm going to add in my daily life physical activity directly into what we call a progress. Secondly, and also very importantly, also what we know now from the literature is that learning more languages has a very profound effect on the decline of neurodegenerations. So I really started a couple of years ago, and already in the past, when I was young in the Netherlands, we learned 4 languages. So I speak, of course, Dutch. That makes sense. I speak English. At least I try to. I'm doing German, and I speak French, and I also have learned now to speak Spanish and Chinese.
That's incredible.
And that's really nice because when you're in a different habit, you can really switch easily and can adapt easily. Secondly, and also very important, is we know also from the literature, but also from our own study, that the first sign that something is wrong in your brain has to do with a decline in your sensory system. For instance, you're losing the capacity to smell. You don't feel pale anymore. Your hearing is going down. Now, basically, you are basically in the capacity. Each individual is in its capacity to work on his own brain. I would like to make a statement that I am my brain. The way my brain functions depends on yourself. If you don't use your brain, you lose your brain. That's an old strategy, but using your brain is something what you can do. So some people rely on the doctor. Some people rely on a pill from a doctor. I would say, forget this. It's better to work on yourself, on your physical activity, on your sensory system. Go out and experience that the sun is shining, that you can smell the grass, that you can feel and touch whatever there is in the world, and increase your sensory information. Because the sensory information, that brings me then back to your locus coeruleus, sensory information is entering the brain in the brainstem. And from the brainstem, there are 3 important areas. You have the dorsal nucleus with the serotonergic cells for depression, the noradrenergic neurons in the locus coeruleus, and the dopaminergic cells in the substantia nigra. They are related basically to anxiety, to motor behavior, and basically to what I would then say more cognitive behavior. So they are interconnected with each other, and they're all related to the information what they receive from the outside world. At the moment, it has also been taught, and that there's quite a good debate at the moment, also in the US, that maybe Alzheimer is starting from the periphery. So it's not starting in the brain, it's from the periphery, and it's basically from what we then call from the gut and from the pancreas. So basically, there's a lot of ideas, but that brings me a little bit to this point.
Yeah, thank you for that illustration. I think we're getting right on the edge of the mechanism of all of these environmental enrichment experiences, learning new languages, as you were saying. I think we're getting a little bit close to how it actually works. You know, when people talk about mechanism, it's the causative elements in a chain of command or a pathway, a biological pathway, for example. You talked a lot about epigenetics at the conference, and one of those things that epigenetics is very influential in is adult neurogenesis. Now, one of my labs studies adult neurogenesis in the hippocampus of mice, but it is kind of a contentious debate. I think the jury is not so much out on neurogenesis in humans, but I would love your take on not only adult neurogenesis in humans, and then how epigenetics is changing in Alzheimer's disease and how those two might be connected.
Okay, so what's the question? How is the relation? I think, and I was this afternoon really surprised when I noticed the comments of the students from our Alzheimer fast track meeting of the various lectures, that my lecture on epigenetics was rather low rated, because some people said, is epigenetics even important in Alzheimer's disease? Question mark. There was a question mark which was made by one of the students. Wow. So basically, if I asked that question myself, then basically I didn't do my job very well at that meeting, because I think, and I really think, that epigenetics is really the trigger towards these problems into the brain. The way the environment is influencing our genetic readout, and by that can influence the brain, is a very important marker. And I will like to stress this more and more to this point. So gene and environment, the genetic part, which is partially available in Alzheimer's disease, but particularly the environmental part, which could be stress, which could be hypoxia, which could be basically pesticides even in the environment, whatever, they do have an effect on the way the DNA has been read by RNA, and by that making proteins, and by that making new neurons. And if you cannot make sufficient new progenitor neurons in the dentate gyrus, you do have a problem, and that problem is called Alzheimer's disease. So I think it's interesting to look in this aspect, and we still have to find more evidence. Well, the first evidence, what I showed at the meeting, and I think we get some good records for that, was that basically methylation, so the possibility to read your DNA, is diminished in Alzheimer's disease in compared to young people like you. And that's an important observation, that we lose the capacity when you get older, when you get demented, to have a sufficient readout of your DNA.
And that's really scary. But I think there is some hope in that exercise is well known to cause epigenetic changes, and most of those are considered beneficial. I think they discussed at the conference, another presentation was 150 minutes. Now, I think we're probably due for a more in-depth explanation of epigenetics in the podcast, but essentially these alterations just open or close the book, like you said, of reading genetic code out. And when that gets messed up, I can see how that might cause problems in the brain. Neurons are very sensitive to the kind of proteins that they can make. But I was hoping to switch gears a little bit and talk maybe a little more about your current research areas of interest. Now, we mentioned the brainstem, locus coeruleus, dorsal raphe, and those monoamine hypotheses, but what else are you very interested in at the moment?
Let me see in the group. In our group, we are interested in the role of finding new therapies for Parkinson's disease. we like to get rid from deep brain stimulation. We used a lot of deep brain stimulation to treat Parkinson's disease, and we are now moving into the field what we then called nanothermal therapy. And nanothermal therapy is the new therapy in which you bring nanoparticles into the blood of a patient. These nanoparticles are transferred to the brain, and with a very specific transcranial magnetic stimulation, we can stimulate a brain area of approximately 1 cubic millimeter. Wow. So a very small area. And why is that important? Because we have used a lot in combination with the company Medtronic's deep brain stimulation. But if you have to use deep brain stimulation, you bring a needle, an electrode, right, into the brain. So suppose you put a needle into the brain, you also disrupt a lot of blood vessels, and you can induce very small hemorrhages into the brain. So at the moment is, if you want to treat a patient, you want to do it in a non-invasive way and not in an invasive way like DBS. So that's an important area which we are really at the moment interested in. And the second thing is that we are really interested in is also looking to what we then called can some cells, some neurons change their transmitter on the basis of epigenetic changes. So can a dopaminergic cell become serotonergic because of a trigger? And this is something which we like to study at the moment a lot as well.
I find both of those personally fascinating, as well as just in general scientifically fascinating, because I do use deep brain stimulation, chemogenetics to be specific. It's a little different than any electrode stimulation that people might think of typically. But I do work with those experiments in mice myself as a preclinical researcher. Sorry, and also the neurotransmitter switching has been a really hot topic, and I think it's in its infancy, but what I can certainly say is I'm very excited to hear more about that. We're looking at possible switching between GABA and acetylcholine, muscarinic.
So in which area?
Yeah, in medial septum there is some evidence that can happen, but not from our lab. Anyways, we're getting far afield. But are there any other things outside of your research area that you're really excited about as well, or in general, perhaps Alzheimer's disease? What are some of the things that got you amped when you heard some of the talks at the conference?
Well, if I look to the various talks, of course, it is quite popular to talk nowadays about neuroimmunology, how immunology is involved, how microglia is involved, what is triggering the activation of microglia, and by that, the further triggering of astrocytes. So that is also an intriguing area to me. Also, in this respect, the gut-brain axis, how is diet has a positive effect on microglia activation. Mm-hmm. Why is a Mediterranean diet and a diet in Japan much more effective than what we call a Western junk food diet? So what is that line? I was thinking, but I will not go into the field of obesitas and brain dysfunctioning. I'm moving a little bit in the field of rare diseases and like Rett syndrome, but also like schizophrenia in relation to Alzheimer's and in relation to neurodegeneration. One area in which I have special attention at the moment is the relation between cancer and neurodegeneration. All kinds of cancer, so the area of neuro-oncology. And I don't mean brain cancers, I mean cancer in general, like prostate cancer, breast cancer, lung cancer, stomach cancer. All these cancers are really inducing an acceleration of Alzheimer's disease, what we're studying at the moment. So this is also a combination which I'm interested in. And in addition to that, a lot of people with cancer are treated with different kinds of drugs. And we are interested at the moment to use these cancer drugs for the treatment of neurodegeneration. This is a nice combination, which is rewarding.
Yeah, that's so many different research interests, and I imagine all of your traveling and language learning is just ramping up that neurogenesis, making you very flexible and plastic mentally.
Well, the nice thing, if you travel a lot, you meet a lot of people. I do have a huge network. I just was checking here by coincidence, my LinkedIn account, and I can see I have about 7,000 people in my account.
Wow.
That's really, and you meet a lot of people. You get a lot of mails and a lot of different requests, a lot of different questions for collaborators. But I really focus on now on some things, and I really like to work with my own team on some very restricted areas. It's nice.
That's a really exciting research career. Makes people want to think twice about how being a principal investigator is supposed to be boring, or perhaps a shut-in, like a hermit job. So definitely breaking the stereotype there, and I appreciate that. One of the things we were really hoping to talk about in this episode was your longtime history with Alzheimer's Fast Track, which is the meeting, of course, we met at. You were the original founder of that conference 16 years ago, is what I'm told.
Yes, I started this meeting basically with an idea, and I think the idea is still valid, and I'm pleased that Bright Focus, but also here in Europe, Alzheimer Netherlands and Alzheimer Germany and France took this over. Basically, I was in the past, about 20 years ago, I was the scientific director in Europe of all Alzheimer related work. And I was the chairman of the research committee of Alzheimer Netherlands, Germany, and France. And in that aspect, we received every year grants. These grants were reviewed, and at the end of the year, grants were awarded. We added to that, first of all, the United States. That was, in that time, it was not the Bright Focus Foundation, but it's called AHAF, the American Health Association Foundation, I think. And they joined us because I really said rather quickly, if you have Alzheimer is not a European thing, it's not an American thing, it's a worldwide thing. So we also should try to bring scientists from all over the world together. That was the first point. And then I mentioned a certain moment that was the most critical moment when I focused on Alzheimer Fast Track. I said, if we get all these grants requests, all these requests for getting funding, but all these grants are written by old people, and everybody was completely upset when I told them to this at that time, and because I was at that time in my 40s, and I said, basically, we have to start to think differently. We have to raise a new class of scientists, of young researchers who will move into the field of Alzheimer and to bring new ideas into the area, and also have a possibility of what we call a sustainability in the research area of Alzheimer. So I said, if we organize a meeting, a 3 days meeting like this is, we would like to go maybe to a 4 days meeting, but if we have a meeting to bring all young people together, who our major task is to make these students enthusiastic for the field of Alzheimer research, then we have the foundation for a new class of researchers who go into the field, who explore the field, and who bring the field a step further. And that was my whole idea, and I think we worked well. It worked well, and in the new grant applications which we see at the Bright Focus Foundation, we can see now also that a large number of young postdocs or faculty members are applying for funding. And I also told the Bright Focus Foundation, don't go for the average, don't go for the normal amyloid pathology, tau pathology. It's better to reward grants which are out of the blue in a new area, a new niche, a new direction, because they are highly expensive, they're risky, and I know most of the organizations, they do not like high-risk projects, they go for low-risk projects, but I said they don't need anything, because in the last 20 years, we haven't made that progress. So the idea was, again, bring young people, train them, focus them how to write the grants and how to, and etc. And that's when it started, and everybody took it over. And in the beginning, we didn't have so much success. Our first meeting was in this respect in Germany. Then we had a meeting in the United States, very close to Washington, D.C., and we started with 20 people. And nowadays, you can see we were this year with 56, and next year we most likely will be with 82 students.
Wow, excellent. And not only the students being there for 3 days, but you got some super high profile PIs, international faculty to agree to come speak and be a part of this. How do you convince people to bring them in? How do you recruit all of that?
Well, indeed a different question. In the beginning, we were really asking people, would you like to join? And basically, the people said yes, but I'm just coming only for my talk. I'm coming in and I'm getting out after 1 hour because I'm going to the Society for Neuroscience. I said, well, we have the meeting is before the society. Nowadays, since 3 years, the picture completely changing. Now people already asking us, would you like us to consider to be a speaker? We are very much interested. We would be pleased if you can join. So the good point is we have excellent people. And since we do have excellent people, other excellent people like to join to meet the other excellent people. So it's really like a self-fulfilling component. And some people say, I'd like to be part of this. It's important.
Yeah, definitely. I was just so glad to be there that there was such a star-studded lineup, including yourself and all of the conference organizers. It just went really smoothly. So I've heard some talk that this was the best one yet, and I hope that they continue to improve in the future. I will sure be there in the future as well. So at what point did Bright Focus get into the Alzheimer's Fast Track? And you mentioned some other European meetings. Are there Alzheimer's Fast Track in Europe that you would want people to know about?
There haven't been any Alzheimer Fast Track, but I know the European organization have also now decided to start a similar program. They asked me to chair that too, and I told them I will do it for 3 years in Europe. And it's going to be starting next year for the first time.
Oh, excellent. Okay. Well, maybe we can follow up and post a link to that when it starts.
In the US, the meeting has always been in conjunction to the Society for Neuroscience. We are at the moment considering if this is important to have this as a meeting. From the students, I received the answer that it's not that important to them. It could also be as a separate meeting somewhere. We haven't decided yet. In Europe, it will be in combination to a big meeting, which we have also in the US. It's called the ADPD meeting, the Alzheimer and Parkinson meeting. And that will be in conjunction to that.
Maybe a conference of interest for myself as well. But I don't want to take too much of your time. First off, thank you so much for coming on again, Dr. Steinbusch. Really appreciate it. And I was wondering if you had anything else you wanted to say to the audience, a lot of which we know are students. I know you're very active in mentorship and networking, so maybe you could give us some tips, perhaps.
Wow, that's a difficult question at the end. Give us some tips. The tips I would like to give to all my students is go for your heart. If you have an idea, go for that idea, because I think that's a very good feeling. Network is important. I always tell to all my students, you have to believe in what you're doing, and you have to sell your science. It sounds a little bit very capitalistic, but it's not the idea. But if you really have to make a good story about what you're doing, it's much easier to bring it forward in a publication and also in a grant proposal. So you really have to believe in what you're doing and sell it in the most efficient way. And the other important thing I would say is that for postdocs and for PhDs, find yourself a team in which you really like to work. And your boss should be from a high standard. And I really think that's important. Don't go for average. Say, I just take a job. I can have a job in whatever, in Kentucky or in whatever. But take a job in a university with a high level and be part of that group and feel proud to be part of that group. I think that's important to me.
Well, thank you so much. It seems to be working out for you terrifically, and I can't wait to see where you're traveling next.
Okay.
Hopefully you can get some rest over the holidays.
Yes, and thank you for your conversation. I enjoyed it.
Absolutely, me too. Thanks for listening, all.
Thanks.
Okay, so some quick takeaways from that episode. First off, adult neurogenesis does happen in humans. I know there's been a little bit of debate about that, but I think it's very well established now, using the best methods and protocols and being very careful. It is, however, sharply decreased in Alzheimer's disease, and that's what Dr. Steinbusch is really interested in, is why that is messed up in Alzheimer's disease and how we can fix it, right? So one of the best things that I think he outlined is that exercise seems to be the critical component in environmental enrichment and going out there and living your life. Go out there, experience new things, try new things, but be active. Keep your body active, keep your mind active, and especially if you can exercise in groups, that seems to be the most beneficial. So if you take anything away from this episode, I know there are a lot of great tips for scientists as well. Make sure you network, make sure that you sell your science properly, but really the exercise component for everyone's health is super critical. So, do that for me. Go out, take a walk, whatever it is. Even light exercise can help. Do it for yourself. And yeah, that's going to be it here for us at the Straight From Scientist podcast. Thank you so much for listening, and especially if you provided feedback or are a sponsor of the podcast. If you'd like to become a supporter, you can go to patreon.com/SFS podcast. Another way to support the podcast is through our affiliation with Plant Warrior. So Plant Warrior produces 100% natural vegan protein, does not contain any whey nor any artificial sweeteners, but it still has all 20 essential amino acids. And on top of that, Plant Warrior pledges to plant 1 tree for every protein bag purchased. It's 100% money back guaranteed, so you can try it risk-free, and in the meantime support the SFS podcast as well as get a tree planted. If you want to support the SFS podcast by buying a bag of Plant Warrior protein, you can use the discount code SFS10 for 10% off. Just make sure that the discount applies when you go to checkout and won't apply in your cart. Thanks all for listening again, and we hope you enjoy each and every episode.